All right, so again, this first one, we're on the little Pac-Man guy, and this is going to be looking at 11.1 and 11.2. Okay. All right, we need to start off with a couple rules, and these rules are for proteins. So these aren't your rules, these are protein rules. So, protein rules. I want you to remember these for like the rest of this class, like not like this class period, but the rest of all of this class, or just the rest of your life. That would work too. These are two very important rules that govern proteins, and if you think about any time you see this occurring, you should immediately think of the changes that are occurring. So here are our two protein rules. Rule number one, the binding / unbinding of a substance and I don't want to go any further than here. Section that off. Yeah, I'm just sectioning it off right there. So the binding and unbinding of a substance to / from a protein will cause here's an important vocab word, a conformational change. Conformational change. What does conformational mean? Conformational change. Yeah, it's form, it's shape. So it causes a shape change in that protein. We already knew this from non-competitive inhibition. The idea that if you bind something to that allosteric site, it can cause the shape of the protein to change, or it can cause the active site to no longer receive whatever the substrate is. Same idea. If you bind a ligand or any kind of receptor to a protein, that protein is going to change its shape. And that is important because rule number 2, changing the shape of a protein will block / open / change their active sites. Remember, the active site is just the portion of the protein that's interacting with whatever it is that you're dealing with. And then there's a little bit more to this sentence. Changing. Vocab word number 2. Their affinity. What does affinity mean? Not infinity, not like unlimited, but what does affinity mean? Yeah, their preference or their desire to interact with, their affinity for other substances. So every time something binds to or unbinds from a protein, that protein is going to change its shape. A shape change in that protein is going to change what it wants to interact with. And this is how you can turn proteins on and off. Like if you want to use like the ideas of on and off, or this is how you would activate or deactivate a protein, is depending on what is binding to or unbinding from that protein. We get specific shape changes that will then allow it to interact with new stuff. And these are the two primary ideas of how this whole signal transduction pathway works. We're going to have a series of bindings and unbindings and releases from proteins or to proteins, which is then going to transduce a signal from outside of the cell to inside of the cell. Because that's really the whole idea of signal transduction. Basically what we have is something is happening outside of the cell, and we need the inside of the cell to know that it's happening and to do something about it, right? We're responding to a stimulus of some kind. The question then would be, okay, why doesn't that receptor, or why doesn't that messenger just go into the cell and directly interact with whatever is going on in the cell. Typically what we see is that these ligands, these signaling molecules, aren't able to get into the cell because of their makeup. Typically they're water soluble, meaning they're polar, and they can't get into the cell membrane. Okay, so this brings us to a definition. We need to define what a ligand actually is. So do that right here. Okay, what is a ligand? So ligands are typically small. They are typically, well, no, not typically, they're always, they have to be a non-protein molecule, so something that is not a protein. They bind to proteins, so which bind to proteins. Causing a shape change, causing a conformational change. Now for us, in our context, so in signal, I'm just going to use signal trans for signal transduction pathways. These are normally water soluble and unable to enter the cell. Oh yeah, for sure. These are typically water soluble and unable to enter the cell. So if they can't enter the cell, how are we going to signal the inside of the cell? Well, they're going to have to bind to something outside of the cell that can then transmit that information to the inside of the cell. And that's the whole idea of signal transduction. Okay. There are 3 major steps in signal transduction that your book goes through. So we're going to look at those individually, give a kind of an overview statement of each of them, and then we'll look at a couple specific examples of the first part, which is the actual reception of that signal. Okay. Everyone ready? No? Man, I already need some work. All right, we good? Okay. Let's move up here. The first thing we'll title this little section. Make it small though here at the top. 3 steps in signal transduction. Okay, and then hug this up to the top as far as you can. The first step that we'll see, switch over here, is going to be reception. And this is covered in 11.2 of your book. Okay. And we will squeeze this all in. Go ahead and make a little barrier here. We'll do all the reception stuff right here. Okay. What we see with reception is that the cells detect a chemical signal from outside of the cell membrane. So this would be some sort of external signaling molecule. Maybe it's a hormone, but it's going to be outside of the cell membrane, and this is going to have to be detected in some way. And it's going to be detected through the binding to a membrane protein. So We'll go just below the picture for the receptor. And we will say that this is caused through a ligand binding to a membrane protein. And this would be our ligand here. This would then be our membrane protein. So that is the overall idea of the reception stage. All right. Next, we would have the transduction stage. This is covered in 11.3. Is everyone good on reception? Okay. I'll leave yourself a little bit more room for this, so see where it kind of goes from light to dark right here? That's where we'll make our barrier. Okay. During this transduction stage, we're going to take that original signal, because remember, when this ligand binds to this membrane protein, what's the membrane protein going to do? Change shape. That change in shape is going to now allow it to interact with new molecules that are inside of the cell, and that's going to relay this signal. So, ligand binding to the receptor protein or membrane protein. Causes a shape change, a conformational change in the receptor protein. All right, and what that's going to do, so then we'll just go directly below where it says relay molecules here. So due to this change, series of events are set into motion which will generate a specific cellular response. And I'm being vague there on purpose. The response that we get is just going to depend on what pathway we're in. We're zooming out and just going over these generic general pathways so that we can then apply them to any given example that we might run across. Okay. Then the final one here is going to be our response. This is found in 11.4. I didn't leave myself much room for this one, but that's okay. Okay, what we see here is the transduction events is then going to trigger some sort of cellular response. So the events in the transduction pathway, so I'll just do trans pathway. That's this pathway. In the transduction pathway, triggers a specific response. Your book is very clear to indicate that this response could be anything that the cell would normally do. So, a couple examples, maybe it's going to cause protein synthesis. So maybe we're going to trigger a specific protein to be made. Maybe it's going to trigger some sort of nutrient release. So your book gives the example of adrenaline being used to release glucose, which would make sense because if your heart rate's going to be jacked up, then we're going to need to get some more glucose in your bloodstream to supply those muscle tissue for the fight or flight response. So there's all sorts of different pathways, or all sorts of different responses that could be at the end of whatever signal transduction pathway we're in. So that is the overall general idea for signal transduction, these three major steps. These are the main things you would need to know from 11.1. So then we move on to 11.2, where they go over three specific protein receptor models. So there are these three major classes of these receptor proteins, and they're all triggered in the same way, but depending on what the transduction pathway is, we can end up with a whole bunch of different responses in them. So the first one that we're going to look at here, and again, this is out of 11.2, and I'm going in order as how your book goes, is going to be called a G protein coupled receptor, which I'm going to abbreviate through this whole thing, the GPCR. You do not want to go past this diagram here. So I'm going to set up my barrier right here. All the way down. And then our other wall needs to be here between these. So we got 3 spots left. All right, all ready? So these G proteins, I can see them being highly confusing. So I want to make sure that we go over how these things work. These are abundant through the living world. We see these all the time. Okay. So here's the deal. These GPCR proteins, they are the actual receptor themselves, and they're going to be closely associated with another protein called a G protein. So they work together. Okay. Let's go ahead and just label them within these diagrams so that we keep them squared away. So this little guy all the way to the left that has like the little V cut out at the top, that is the GPCR. The round little guy that has the GDP on it, that is going to be a G protein. So that's the G. So just go ahead and label that in each of these, just so that we keep these square. So they're going to be working closely together. All right, here we go. So, first of all, clarifying idea, GPCRs. So G, protein, coupled receptors. R, protein, receptors, which work closely with G proteins. Here's how they work. Yes, for sure. So I'm going to number these events, and then we'll label them on the actual diagrams themselves. Okay, so inactive G proteins contain a GDP molecule. Okay. I originally was going to write this on the sketch note, but I don't think I need to, because I'm just going to tell you this now, and you're going to remember it. Okay. GDP and GTP, what does that kind of sound like? ADP and ATP, right? Okay. What is the A in ATP? adenosine, which is a form of adenine. Adenine is one of the base pairs in your DNA, right? A's go to T's, G's go to C's, right? Okay. The G is guanine in this. So GTP is guanosine triphosphate, and GDP is guanosine diphosphate. It's the exact same thing as ATP, just with a guanine instead of an adenine. Okay? So don't let that throw you off. Basically, it's just a source of phosphorus, source of phosphate groups. And phosphate groups can be used to turn proteins on and off by phosphorylating and then removing those phosphate groups. So this is just a source of phosphate. Ooh, I bet it exists. I'm sure they all exist. I just don't know what they're used for. Yeah. Anyway, so that's the first thing we see is we have these inactive G proteins. They are not going to be able to do anything. And we see that here. Here's our inactive. So it says G protein inactive. So there's our inactive G protein with the bound GDP. So in order to get a signal relayed, we're going to have to bind a ligand and then activate this G protein. Because this guy's going to serve as like a middleman between the GPCR and then whatever enzyme or other protein is going to be used in the pathway. Okay, so then step 2. It all starts with a ligand binding. So when the ligand binds to GPCR, a shape change occurs, and that shape change is going to open. So a shape change opens a binding site for the G protein on the GPCR. So again, whenever proteins change their shape, their affinity for other things increases or decreases. In this case, we're going to have a ligand binding. So GPCR right here, ligand binds. That is then going to open up a site on the GPCR that increases its affinity for the G protein. G protein is going to slide over and bind with that GPCR. So step 2 then, I might have come off that too fast. You guys get on here for number 2? Okay. So then step 2 is happening right here. Okay. Then number 3. All right, since the G protein just bound to something, what's going to happen to the G protein? It just latched onto something. No. Anytime something binds to or unbinds from a protein, what does that protein do? Changes its shape. Yeah, so now the G protein is going to change its shape. Okay? So the binding of the G protein changes its shape and releases the GDP. When proteins unbind from things, what do? They change their shape. And since it has released GDP, the G protein is now going to change its shape once more, which is going to make it want to pick up a GDP or GTP. So many abbreviations. Okay. So it's going to release the GDP. Then it binds to GTP, activating it. And by activating it, what does that mean? Well, it just bound to GTP, so what's going to happen to it? It's going to change its shape, right? Which is going to allow it to interact with something new. Okay, so we see that happening here. Here's step 3. So here's what happens. The The G protein has this bound GDP, it's inactive, right? The ligand binds to the GPCR, changes the shape of the GPCR to then accept the G protein. G protein slides over, binds, changes its shape again, which causes it to release the GDP. Once the GDP comes off, it changes its shape again, and it binds the GTP, because its affinity for all of these things has been changed every time its shape changes. When the GTP attaches, it changes its shape again. And now it is going to be activated and ready to interact with some specific other molecule. And in this case, it's going to be this enzyme over here, which is currently inactive. How do you think we activate this enzyme? By binding the G protein to it and changing its shape. Right. Okay. So that is the final step for us in the figure. The G protein is now going to be able to bind with a new molecule. So the activated G protein can bind to new molecules, relaying the original signal. Okay, so this again is just the reception stage. This is getting from a ligand binding to the very beginning of the signal transduction pathway. As soon as the G protein interacts with a new molecule, the signal transduction pathway is going to start. So this arrow right here is showing that new signal transduction pathway that's going to cascade through the cell and eventually elicit some sort of response. So 4 is occurring right there. Then after the G protein binds and this enzyme does its thing, it's going to kick off the G protein and the whole thing resets. But we don't need to go through that resetting stage. So that is the GPCRs, the G protein coupled receptors. So there's 3 major examples your book used. The other major example that your book uses is this next one over. I know this is a lot. We're just going to try to plow through this thing. Okay. This schematic right here is showing something called a receptor tyrosine kinase, which I'm going to call an RTK. Receptor tyrosine kinase. Right. There's 2 major parts to these things. So look at the very top left drawing of this. You can see the cell membrane. You see 2 different proteins, and they're not stuck together. This is the inactive form of these tyrosine receptor kinases. The receptor part is out here. So I'm just going to label that R. It's outside of the cell, and it's going to be receiving the ligand. The tyrosine kinase is the part that's inside of the membrane, the TK, and that is the region that contains the kinase. Remember, kinase is an enzyme that adds phosphate groups to things. And tyrosine, TYR, is an amino acid. Okay? And this amino acid is bound to the protein. It's just hanging off. And these are going to be receptor sites for phosphate groups. Okay. So let's go ahead and define what a kinase is. Okay. So a kinase is a class of enzymes. So it's going to be an enzyme which catalyzes, meaning it speeds up the transfer of the phosphate group. So it's going to be taking phosphate groups off of one thing and sticking it to another, essentially. This specific kind of kinase, so the RTKs, are membrane receptors. So mem recepts, which transfer phosphate groups from ATP to bound tyrosines, which are, I'm just going to put AA for amino acids. So a tyrosine is one of the many amino acids, one of the 22 amino acids that we see. Okay, so let's look at how this works. So first thing that's going to happen, just like with all of these, is we're going to have a ligand binding to that receptor site. So ligand binds to the receptor bringing the two individual, we're going to do polypep for polypeptides, two individual polypeptides together. And this forms what's called a dimer. A dimer is just whenever we have these two individual proteins come together and form one overall protein. It's kind of like going from two proteins in tertiary structure to a single protein in quaternary structure. Dimer. It's actually labeled somewhere on there. Here it is, that second one. Dimer. And actually that is where we will put our number one. We see the binding of the ligand. When the ligand binds to the protein receptor, what does the protein receptor do? Changes its shape. When it changes its shape, they will have a stronger affinity for each other, and that'll cause them to stick together, forming the dimer. Once they stick together, what are they going to do? Change their shape, right, and that's going to cause them to change their function again. And that brings us to number two. Dimer formation. This one has 4. No, this one has 3. 3 and then a final statement. So we've like 4 lines. So dimer formation activates. So we labeled earlier the R region and then the TK region, so the receptor region and then the tyrosine kinase region. So everything underneath the membrane. So it activates the TK region. By activates, again, we just mean that its shape changes in a way that now it can do something new. So it activates the TK region and a phosphates from ATP transferred to the bound tyrosines. We're basically just creating like this little bank of phosphate groups that can be used for multiple pathways. The cool thing about the RTK pathway, these tyrosine kinases, is that you can cause a whole bunch of different pathways to occur at once from one individual ligand binding, because these phosphate groups are not specific to any one pathway. So a whole bunch of different transduction pathways can be set off from just one signal, from one original signal. Okay. That is occurring up here, right here in the middle for number 2. So the dimer forms. Now we're going to have phosphate groups that get stuck to the end of the tyrosines. You can see the P's that are circled. That's indicating the phosphate groups. Okay. And then that takes us to our last step, but then leave one little strip at the bottom. The last thing that we'll see is that relay proteins. So relay proteins are going to be the very first protein of the transduction pathway. So So they're going to take these phosphate groups and then start doing stuff with them. So relay proteins are phosphorylated. And activated. Again, activated just means a shape change, allowing it to do something new. Setting off a transduction pathway. And here's our wrap-up statement on this one. This is important because we can get multiple pathways from the same ligand. So you can set off a whole bunch of stuff happening at once with the release of like one hormone, for instance. You get a bunch of stuff happening. Okay. Our very last one, I know we get out of here in 3 minutes, but we actually did it last class period in exactly 3 minutes, so we can do this. And you're right here in this little corner. Okay, you ready? Here we go. So number 3 is just going in that last box. Okay. This thing is called a ligand gated ion channel, which I will be calling the LGIC. LGICs are, I'm just going to do recept for receptors, which serve as gates. I'm going to put gates in quotes because it's not really a gate, it's just a blocked channel for various ions. We only have one step here. And I'm accidentally doing it in the wrong color, but that's okay. So ligand binding, we're going to say it opens the channel. We know better though. It doesn't actually open the channel, it just changes its shape, which allows these ions to now move through the channel. So it opens the channel, allowing ions to enter the cell. This is normally used by your nervous system. Used to generate electrical impulses. And the middle picture, so there's the top, the middle, and the bottom. The middle picture is getting the one, because that's where it's actually happening. So this is very similar to what we saw with non-competitive inhibition. It's just backwards. We're activating a protein instead of deactivating a protein. We have an inactive channel, it's blocked, ligand binds, channel is unblocked, ions fall through. Typically these ions are going to be negatively charged, a lot of times they're chloride, and that big free fall of negative charge will shoot off an electrical impulse, which can be used in your nervous system to do all sorts of stuff, to send signals. So these are the major types of signaling and reception that we see. These are the ones that are addressed in your book in sections 11.1 and 11.2. And tomorrow, I know this is grueling, but I feel like this is the fastest way to go through an entire chapter. Really, we're like doing an entire chapter's worth of stuff in 2 days. It's pretty good. Impulse, electrical impulse. Tomorrow we'll do 11.3 and 11.4, which is transduction pathways and responses, which I haven't finished writing yet, and I'm really hoping that it isn't super, super long. I can do it in one day.